It's my great pleasure to introduce today's second speaker, Siqi Liu. She just started as an assistant professor at UT Southwestern Medical School about 8 months ago. And Siqi started her scientific journey at University of Science and Technology of China as an undergraduate student, where she was working on characterizing a new gap that controls cell migration. She then moved to United States and did her PhD study with Zhijiang Chen at UT Southwestern, where she was really deepened our understanding about IRF3 activation and recruitment for host defense, as well as some of the ubiquitination mechanism for antiviral signaling pathways. Both works are extremely well characterized in the field. A lot of people actually immediately recognize those are Siqi's work when I was talking to them. So Siqi then performed her postdoc with Elaine Fields at Rockefeller University, where she's going to tell us about today on the immune and epithelial cell crosstalk. She has won a lot of prestigious fellowships, including the John Coffin Child's Postdoc Fellowship and K99 Art00 Awards. As an independent researcher, as an assistant professor, she has already also won multiple grants, including the UT System Rising Stars. So her lab is actively recruiting technicians as well as postdocs. So definitely reach out to her. And we are really excited to host the Siqi to tell us about her postdoc work related to injury repair, a host response to homeostatic disruption. Welcome, Siqi.
All right, thank you so much, Xiaoli, for that very nice introduction. Can you hear me clearly?
Yeah, we can hear you.
Okay, great. So I guess I'm very excited. Thank you so much for Fragile Nucleosome to invite me for this talk. And I guess my study will be like let you to take a break from the chromatin field, but just to rather give you overview of how actually I came to understand this injury repair mechanism, but in the end may lightly touch upon that how we understand at the transcriptional level by just looking at some targets. But it's gonna be very brief. So basically, I wanted to understand how our body sense injury and repair it. And we understand this problem as a general host response to homeostatic disruption. The ability to maintain homeostasis is essential for life. So our body is maintained at a homeostatic condition at cellular level, tissue level, and organism level. And these are some examples for homeostatic regulation. However, we constantly experience disruption of homeostasis. This will actually require conserve the mechanism to recognize such disruption, then mount appropriate response for us to return back to homeostasis. So, for example, pathogen infection and tissue injury are two universal and ancient disruptions that almost all organisms has to constantly face throughout their lifetime. Well, we know a lot better about how pathogen-induced non-self molecular patterns are recognized by the host, which then generate cellular response to leads to production of cytokines that orchestrate host defense. We know less about how exactly we recognize injury, which then leads to the host response that are appropriate for repair. But this process, they share common features. For example, they are both acute and they have to be resolved for host survival. They lead to cytokine release, systemic inflammation, and so on. And they also have common risk factors, as we know that as we age, we have declined ability for pathogen defense as well as healing our wound. And they all have genetic predispositions. Well, when I was as a grad student many years ago, I devoted to identify molecular signaling pathways that recognizing pathogen-induced non-self patterns, and which then leads to this famous interferon pathway activation and activates transcription factor state 1 that is essential for host defense. As I transitioned as a postdoc fellow in the lab of Elaine Fuchs at Rockefeller University, I switched my focus trying to understand how do we sense injury and how does this injury induce host repair. And with that, we use mammalian skin, and we consider this is as a pretty good model to study tissue injury repair, as skin is the outermost layer that isolates us directly from the environment. And it's more like our innate immune system as a first layer of defense. So to simply put, to induce a wound, we just remove a piece of skin from the mouse back. And then when you have an open wound occurs, the epithelial cells here will start to proliferate and migrate through a process of re-epithelialization to close the wound. And at the same time, the dermal supporting cells will also accompany this re-epithelialization process to rapidly undergo proliferation, angiogenesis, which will help eventually for the wound closure for repair process. So our skin has very robust ability as we constantly get injured and it has to be repaired. And this skin system has a representative structure for most epithelial organs, so we can study generalized mechanisms. And also importantly, when you have a wound, it actually generate this localized response where you can clearly recognize where the injury bed is, where the surrounding healthy tissue is, and we can study this process in time and scale, and exactly we can understand where the tissue and the cells coordinate with each other. And we also has very powerful mouse genetic tools to study skin epithelial specific in a specific manner. So today I'm gonna talk to you roughly about two stories that lead us to a better understanding of host response to injury. So first, we identified a crosstalk between the local epithelial cells and the local T cells. So upon this, we identified the essential role of a transcription factor STAT3. So when we went upstream in the epithelial cells, and we also identified the pathway upstream that directly sense damage for repair. All this started with trying to understand how age decreases the ability for wound healing, as we know that age individuals has lower ability for wound repair. So we compared the wound healing ability in the young versus aged mice. As you can see, this is a sagittal section of a skin wound. In the young animals, the wound re-epithelialized as highlighted by the green keratin 14 staining of skin epithelium. This is re-epithelialized rapidly. And in the aged animal, this process is greatly delayed. So we first probe for the transcriptional differences between these two processes. So we actually purified and isolated the epithelial stem cell, which is the basal cell near the injury bed, and also compared with the homeostatic condition from both young and aged animals. So this is the transcriptome analysis. Just want to highlight to you that, so under homeostatic condition, you have young animals have these genes that are minimally expressed, but these are cohort of genes that are rapidly upregulated in the wounded epithelial stem cells. However, this wound-induced upregulation of these cohort genes are not really upregulated in the aged animal. So we looked closely to see what these genes are, and the cohort of genes are important for immune function. Especially, we are very excited to see a family of molecules named skins. So they are greatly upregulated in the epithelial stem cells in the young animal wound edge, but they are not upregulated in the aged. The question is, are skins important for wound repair? So we use this ultrasound-guided in utero lentiviral injection to introduce shRNA against each individual skin genes, so into the amniotic sac of E9.5 embryo of mice. So this lentivirus will only infect the first layer of epithelial cell progenitor at that time, which will later derive into the entire skin epithelium. So this is a robust method for us to dissect epithelial-specific gene regulation. So we found that skins are a family of molecules comprised of 13 members. So we tested each member, and we found that when we knock out a family skins named skin 3 versus a skin family member of skin 9, they greatly decrease the ability for the wound to re-epithelialize. So now the question is, so wound-induced skin subregulation. So what would be this wound-activated transfection factor that is important? So we pull out all the promoter regions of this skin gene that are upregulated in the skin wound epithelial cells, and we did a motif analysis of the promoter region. So one transcription factor that really highlighted and presented in all these skins are STAT3. So we then tested, we generated skin epithelial specific STAT3 knockout, and we found that so all these skin genes that are upregulated in the skin epithelial stem cells are indeed downregulated if we knock out STAT3. So this suggests an essential role for STAT3 to upregulate these skin genes. So next, we also look at whether STAT3 is activated in the wound edge. So as these are sagittal sections between young animal and aged animal skin wound edge. So this is the wound edge, and the dotted line indicates a border between the epithelial cells and dermal cells. You can see that in the young animal, after 3 days of wounding, you can see robust phosphorylated STAT3 being present in the nucleus of these epithelial cells, but they are not really present, they are really weak in the aged day 3 wounds in the epithelial cells. So suggesting there is really a dysregulation in the aged animal for STAT3 activation. So basically we present a pathway that won't actually induce epithelial STAT3 activation, which is important for skin subregulation, and this is important for repair. So what does skin regulate repair? I'm not gonna go into the detail, but rather summarize this part. So skin's molecule, a family of surface molecule that are structurally similar to PDL1, for example, like immunoglobulin protein, for example, PDL1, CD80, that are famous molecule to regulate T cell functions. So we do have evidence showing that it regulates the activity of a subset of intraepithelial gamma delta T cells. And these intraepithelial gamma delta T cells, all named DTCs, they are also important for skin wound healing, as proposed before. So basically, we found a signaling cascade that is upregulated in the young wound edge, and this cascade is diminished in the old wound edge, which kind of suggests that they could be one of the mechanisms that why aged individuals has delayed healing. However, during this process, we actually found a very, which I think is very phenomenal phenotype, is that so when we knock out STAT3 in the skin epithelium, it actually completely halted wound epithelialization, which is more dramatic than just knocking out skins or gamma delta T cell per se, which suggests that skin STAT3 as a transcription factor actually regulates more aspect of wound repair than just modulating the downstream skins and gamma delta T cell alone. So this is just to give you a flavor of how the STAT3 knockout look like. So this is a Y type. You can see that green labels the skin epithelium. It re-epithelializes with a time course of 5 to 7 days. And when you delete STAT3 in the skin epithelium, specifically, it greatly halted the re-epithelialization process. So this really suggests that STAT3 is one of the master transcription factors that are induced by injury. It regulates host repair. But this actually reminds me back in my PhD study where we study how pathogen-induced non-self patterns induce master cytokines like type 1 interferons, which then activate STAT1 that is important for host defense. So STAT1 and STAT3 belongs to the same transcription family factor. So I was curious whether from what our data look, the role of injury-induced STAT3 activation and its role in host repair really analogous to the role of pathogen-induced STAT1 activation in host defense. So the questions, are they analogous? Then first we want to understand how does the injury induce STAT3? So basically I took another mouse skin, I isolated the wound and I separated from epithelial compartment and the mesenchymal dermis compartment. I went to search for known STAT3 activating cytokines and their presence in the wound. Remarkably, there is one cytokine really jumped into our eyes. So this cytokine is named IL-24. So IL-24 is remarkably not expressed in anywhere in the homeostatic tissue, but it's rapidly upregulated in the skin epithelium. So we have evidence showing that IL-24 is specifically expressed in the wound edge epithelial stem cells. And is IL-24 essential for wound repair in the wound? So the answer is yes. So the phenotype is not as dramatic as STAT3 knockout, but actually has a very pronounced phenotype. So although here, so this is a comparison between, we generated IL-24 knockout using CRISPR-Cas. So this is a comparison between the IL-24 wild type versus knockout. So as you can see that this is a day 5 wound edge. The green marks the epithelial cells that are still going into the wound region as they are migrating into the wound. So remarkably, complying with this epithelializing migrating epithelial cells are a robust dermal repair response. So the gray dots indicates proliferating cells labeled with EDU, and you can see that there are robust. So magenta indicates endothelial cells, that indicates there's a robust angiogenesis. So basically, there are multiple dermal cells that are proliferating and accompanying this migrating tongue. However, when we delete IL-24 knockout, you can see only a thin layer of epithelial cells is still migrating, but this dermal accompanying response is abolished. So suggesting this epithelial-derived cytokine actually coordinate a response between the epithelial compartment and the mesenchymal compartment for a coordinated response. So we also have evidence showing that IL-24 and the receptor are essential for dermal repair, and if we use another genetic to mediate by the Sleeping Beauty transposon-mediated epithelial-specific deletion of IL-24 also phenotype copies this effect. Now, so we suggest that injury actually induce a cytokine called IL-24, and IL-24 induce STAT3 activation, and this is actually important for a coordinated pro-angiogenic tissue repair. The question is, what injury signal induce IL-24? So we first wondered, given the analogous pathways between the pathogen and the injury, whether when you have a breach of the barrier, there will be pathogen infection that causes upregulation of the pathway. So our analysis suggests it's not, so IL-24 expression is not dependent on germs or any innate immune adapter proteins that are mediated by that are mediating pathogen infection induce the pathways. So suggesting this is really a steroid injury induced molecular pathway. The question is, what pattern really induces? So then we back to our drawing board and thinking about our paradigm. So basically we hypothesize that when you have disruption of homeostasis, it will generate abnormal patterns that normally don't present in the homeostatic tissue. So these abnormal patterns can be recognized by the host tissue, which then produce effector proteins that are aimed for resolution. And whatever it's trying to resolve is against this original abnormal pattern it detects. So given that we find that, so the IL-24 effect is on the coordination of a angiogenic tissue repair, we asked so whether this pattern that is trying to sense this pathway is from some sort of blood vessel disruptions, as when you have a wound, there are blood vessel disruptions, and it cuts off many nutrients and oxygen, for example. So basically, we established the in vitro skin epithelial culture that trying to mimic in vivo wound edge. And we test the panel of nutrient oxygen deprived conditions that mimics the nutrient deprivation status by blood vessel disruptions. So just to give you an overview that we test, for example, lack nutrients, lack oxygen, and alternative substrate or glycolytic product oxidative stress. So among all these patterns we tested, so one thing stand out, that is hypoxia. So basically what we found that among all the patterns we have tested, the hypoxia is the only one that really robust induced IL-24. But want to also mention that, so basically from this cultural system, robust IL-24 induction is dependent on number one, hypoxia, and number two actually depends on the IL-24 receptor. So IL-24 receptor, including both receptors, are highly expressed in the wound edge epithelial stem cells in vivo, which somehow stopped expressing in the culture cells. So we had to reconstitute them. So basically there are two factors. I'm not going to show you the data, but we also have evidence showing that wound bed is highly hypoxia due to blood vessel disruptions. And when the area at the very front of wound edge where you don't have blood vessels is very hypoxia. And this correlates with where IL-24 is produced. So next, we want to test two transcription factors. So first, HIF1 alpha is one of the master regulator that are induced and activated by hypoxia, and we want to know whether that is important. And another thing is that IL-24 acting on the IL-24 receptor will trigger majority a STAT3 activation event, and we want to know whether that is important. So we still use a cell culture experiment, and then we cultured K14 inducible HIF1 alpha conditional cell lines to basically tamoxifen treatment will block delete HIF1 alpha, functional HIF1 alpha. So as we found that this robust IL-24 induction induced by hypoxia is completely abolished by knocking out HIF1 alpha, showing here. We have evidence showing that HIF1 alpha is important for wound-induced IL-24 in vivo, and also we have additional evidence showing STAT3 at the same time is also important for robust IL-24 induction. So this is almost the only piece of data that I have related to this audience. So basically, we are trying to understand how does these two transcription factors influence IL-24 expression, whether they directly bind to IL-24 locus or not. So basically, we first look at RNA-seq data showing that IL-24 is not expressed in a normal homeostatic epithelial cells, but it's rapidly, robustly upregulated. And ataxic showing that there is chromatin regions around the IL-24 locus being rapidly opened after wounding. And then we did cut and run-seq for using IgG as a control. So we tested both HIF1 alpha and the STAT3 binding. So this is actually in the tissue culture, but you can see that upon hypoxia, the HIF1 alpha and the STAT3 binds to these 5 open chromatin regions that identified also in the wounds. So suggesting that HIF1 alpha and STAT3, they both directly binds to IL-24 locus and possibly regulate its expression. Now, so we identify the pathway that is induced by the steroid injury. That, for example, one of the trigger that we found is tissue hypoxia, could possibly caused by the blood vessel disruptions, which then stabilize HIF1 alpha in the epithelial stem cells. This is important for IL-24 expression, and IL-24 will then act on the receptors to activate STAT3 signaling, but STAT3 will actually go back to feedback on the HIF1 alpha IL-24 production part. And this feedback loop is kind of seen also in the STAT1 regulated interferon response. So this will collectively regulate pre-angiogenic tissue repair. One more point I want to make is that, so now you have, say, this paradigm that they look very similar. The question is, are they not analogous, or they are actually homologous pathways? So in collaboration with the evolution biologist Qian at UT Southwestern, we bioinformatically extract all human genes that share homology with L24. These are about 59 secreted molecules. And as remarkably that we found so the L24 family containing a few members sit highlighted in the yellow, sits right next to the interferon family, highlighting in orange in the phylogenetic tree, suggesting they share a closer common ancestor compared to the rest of the molecules. So we also have evidence showing that L24 receptors are close homologs to interferons as well. So this actually suggests that more important paradigm that I think that these are not only analogous pathways, they are actually homologous and common ancestral pathways to resolve disruptions given their multiple level of sequence and structural homology along the pathway. So first we have pathogen versus injury that both can generate patterns that normally don't present in a homeostatic tissue. And then they induce cytokines that are homology to each other, interferon versus L24, and then they act on receptors that are closely homologous to each other. And these also induce homologous transcription factors, and being when the function of when is being for the first line of defense, and the pathway identified here can be served as the first line of repair. So in addition to that, I want to highlight that this study also highlights an essential role of epithelial cells as the first line of defense and repair, as traditionally we think neutrophil and other innate immune cells are the major first responders. So then they directly sense pathogen and damage and orchestrate defense. So hypoxia is actually one pattern, but it's not everything. So basically, in the future of our lab, we will continue to study like how other steroid injury-induced patterns induce this pathway, and also there's other cytokines. For example, another homolog that is IL-19 is also induced by in a different part of the wound bed, and what would that function be? So with that, I would like to thank my postdoc mentor and Elaine Fuchs and the Fuchs Lab members who has been very collaborative and also insightful in helping me with this study, and also my co-mentors at Rockefeller University. So my fantastic collaborators, including Xin with hypoxia, and Qian for the cytokine homology study, Angelina for germ-free mice, and EM study from Milia, and also bioinformatics from Jidu. So my young lab is right now at UT Southwestern is growing. So I have 2 lab members, and we are still looking to recruiting talented grad student, postdoc, or research techs. So this is my lab website, and also thank you for my funding source. Thank you so much for your invitation, and I would like to take any questions.
Thank you, Siqi, for the wonderful talk. And so there are quite a few questions. I think Christine have some questions that she would like to ask herself.
Sure. Yeah. Thanks. Thank you for the talk. That was really cool. I was curious because I think about aging a lot, and you talked about it a bit in the beginning. I was wondering, is the IL-24 expression lower in the aged animals too? Sorry if I missed that, if you wrote that or said that.
That's a very good question. So so far we found out that the MR level is not altered. So we suspect that either it's a complementary pathway, for example, IL-19 pathway might be altered, or the receptor for receptor function during that may be altered. So, but the IL-24 MR is not altered. Yeah.
Okay, cool.
Yeah.
And then kind of just like a more broad question, because I don't know much about skin wound healing. Is the stem cell niche, does it get depleted in aged animals too?
Yeah, so they have lower ability. So there are contradictory reports, but overall, it depends on what niches. So the endothelial stem cells are fine, but hair follicle stem cells are kind of depleted. So they could be also contributing. Their intrinsic effect could also be contributing with the aging. Yeah.
Cool. Thank you so much.
Thank you. Yeah, so I was wondering, like in your homologous pathway, it's like stat 1, stat 3. Given they are homologs, I would think they are going to be very similar to each other. So do you think like in this homologous pathway, it's the transcription factors like very different from each other, or they are actually redundant? It's just they get activated, like it's just they are expressed differentially.
Yeah, so basically this transfection factor, they look very similar, but they actually bind to very different targets right now. So basically, for example, in the Drosophila, there is only one stat, and then to human, there are 6 stats.
6.
Yeah, so they are all kind of induced by different stress response. So basically the pattern or like their function are homologous, but what genes are regulation is really specific to that initial trigger. So from this study actually would propose that, so basically there is an ancient organism that all these only share a common ancestor. So there is a common ancestor pathway that is induced by either pathogen or maybe tissue injury most likely, and then later it diverged into separate pathways to be able to deal with more specific initial insult. Yeah, right.
Yeah, that's really interesting. Yeah. So Gamon has questions wondering like what is the role of the senescence in wound remodeling, and whether the same cascade is needed for wound healing after radiation therapy skin damage.
I think that's a great question. So personally, I haven't really investigated the senescence in skin wound healing. So I mean, there are a few papers showing that senescence is mediated by SIGA-STIM pathway, for example. That is more like the pathogen defense pathway, but they could be interacting with our pathway. So that requires further investigation, for example, whether senescence cells actually induce either IL-24 or IL-19. I think that's a great question. Yeah.
I guess in the patients, like, do you see any mutations in IL-4 gene that just bypass the need for HIF-1 alpha or STAT3 binding?
So you mean like a STAT3 activating mutant, something like that?
Yeah, or IL-24 promoter locus. Yeah.
Right. So for example, I think that there are STAT3 dominant negative mutations has been identified, and this patient does have skin inflammation problem, that they have hyper-thickening of the skin that resembles either a cirrhotic lesion versus a wound healing. They kind of look similar to, yeah, so there are some pathways has been identified. And we are also trying to identify human mutations for IL-24 versus IL-19, for example.
Yeah, that's really cool. Yeah, thank you, Siqi, and thank you both for the great talks, and really hope best of luck of your future lab, and we look forward to more studies coming out from your lab. Yeah, thanks everyone for joining.
Thank you so much. Thank you everyone for listening. Bye.
Bye. Bye.